Thanks so much, Sam, and thanks to all of you. It's really a pleasure to be here and get to talk about diatoms. Does anyone know what they are? Couple? Well, okay, I hope by the end of this you'll have some understanding of what a diatom is and how you can use diatoms to address environmental problems. And so the topic of my seminar today is really going to revolve around three things. These organisms, diatoms, lake sediments, and nitrogen, particularly nitrogen that comes in the atmosphere. So I hope to be able to tell you a story that links all of these together. And I want to show you how one of the ways that we use diatoms to understand what's going on in the environment. And I'd like to echo and welcome anyone who'd like to come and join afterwards. I'd be glad to talk about biology or, career stuff or whatever anybody wants to talk about. So please join afterwards. So nitrogen first. I'm going to start off with that. Nitrogen is an important element. It's an element that is important in controlling composition, diversity, dynamics, really the functioning in a number in all ecosystems, whether it's marine or freshwater or terrestrial. And this nitrogen can be emitted in reactive forms, not as N2 gas, but as nitrite, nitrate, and ammonium. And those amounts of nitrogen that are being emitted in a reactive form have really increased in the last 100 years. And this plot shows some important things going on of what human impacts are doing. We know population is going up. The amount of carbon being emitted. And so this carbon emissions over here, you can see that carbon emissions are going from increasing, I don't know, maybe 5-fold. Carbon dioxide is going up. Nitrogen is going up along with human activities. But if we look at this amount of total reactive nitrogen, it's not 5-fold or 10-fold, it's going up nearly 200 times. And nitrogen is not a greenhouse gas, so it doesn't contribute to warming, but nitrogen has some other impacts. And so I'm going to talk about those impacts and how they are changing places that are particularly sensitive. Okay, now diatoms, just hold on if you don't know what they are. I'll get to that in a little bit. But these things, these diatoms are changing in their composition, so the species, and in how productive they are in low nutrient. So oligotrophic means in low nutrient lakes across the western US. So this is a regional pattern. And we think that these changes that are taking place are in response to nitrogen. They're not in response to temperature. And that's the story that I'm going to tell you. And how are we coming up with this story and being able to interpret it? Okay. So the first part of it is nitrogen comes in terms of wet fall and dry fall. So it's emitted by different sources, primarily close to urban areas, from agricultural areas, and feedlots, large. There are a lot of sources of human introduced nitrogen. And that nitrogen can come out of the atmosphere as in a number of different ways. And one of the things that it does is this map shows how much is deposited in kilograms per hectare per year for some of the western states, Idaho, Montana, Wyoming, Utah, Colorado. And here the blue is 2 to 3 kilograms per hectare per year. And this red is the higher concentrations of 3 to 4 kilograms per hectare a year. And if you know your geography of the West, you'll see that, the places where the nitrogen is really very high are the places where there's mountain ranges. And so why is that? Why do you get more nitrogen coming in mountain ranges? And if we blow that up a bit and look at Grand Teton National Park, we'll see it really is on the tops of those mountains. And it is really a orographic effect. So the mountains cause precipitation, rain and snow to fall on them. And so they're the areas where there is the highest precipitation, and that also brings out the highest concentrations of nitrogen onto or the highest totals, I shouldn't say concentrations, the highest total deposition onto those areas. So not only do they get the highest amount, they're also the most sensitive. And so these diatoms, we're going to look at them and say, are there diversity and production in these lakes being influenced by this atmospheric deposition of nitrogen? So what are they? Okay, so what are diatoms? I'm going to talk about that. I'm going to talk a little bit about lake sediments. And I'm going to talk about how these two are linked, of diatoms, lake sediments, and nitrogen. Okay, diatoms are algae. They're single-celled. So algae are this really heterogeneous group of organisms. It's kind of like saying bugs. There are many different kinds of algae. They're not necessarily closely related to one another, but diatoms are single-celled. And what is the big deal about diatoms is that their cell wall is made of silicon dioxide. So they've got essentially glass cell walls. These cell walls, so they're inorganic. They don't decompose after the death of the cell. They simply sit there. And that is one of the things that makes them really nice as environmental indicators. And they're photosynthetic. And that's important too, because that relates into how they respond to things like increases in nutrients. They have a lot of other features, but all you biologists will understand those, but we don't need to go into detail about that right now for this part about the application. So, so diatoms are photosynthetic, and this is not actually a diatom. We can't see any, but this is an aquatic macrophyte, but it is covered with diatoms, and the diatoms are taking up CO2 and generating oxygen, growing on other aquatic plants. They are very important organisms. Here is an image of a marine diatom. This is a chain-forming diatom. And diatoms produce some 30 to 40% of the Earth's oxygen through photosynthesis. And so here are these organisms that a lot of you have not even heard of before, but say every third breath you take, every third inhale is coming from oxygen that is produced by the diatoms. So they are extremely relevant, even though you don't see them. Here's a picture of a live diatom, and here's its glass cell wall. You can see the outline of it, the nucleus of the cell. They have a characteristic type of chlorophyll A and C. This is the chloroplast. And then they also have a high production of lipids. They are very good energy sources for the zooplankton and higher level organisms that feed on them. And if you take some diatoms and you get rid of all the organic material and you just leave the silica cell wall, they have these incredible patterns in the silica cell wall. And that's what the taxonomy is based on. They're based on this ornamentation. Here's a couple of cells in a chain, in a colony. Here's a different species. Here's some of the silica cell, the bands that keep the parts of the diatoms bound together. So this is what happens when you take some sediments out of the bottom of a lake, whether it's these Teton lakes or Utah Lake, like Sam works on, or Great Salt Lake. You can see these organisms, and they accumulate in the lake sediments. And lake sediments are really interesting vessels. Lakes are out there taking in. They're acting as sentinels. They are watching what's going on, and they record it in the sediments. So lake sediments are going to record what's going on in the atmosphere, in the watershed, and processes that go on in the lake itself. And so what sort of things are those? What does that mean? We can have things coming in the watershed, all sorts of inorganic minerals, nutrients, anything that's washing off of the watershed and ending up in the lake. We can have atmospheric inputs, and that includes anything that falls out of the atmosphere onto the lake itself. Spores, charcoal, volcanic ash, radioactive nuclides. What I'm talking about today is going to be this wet and dry deposition, this nitrogen that ends up in the lake. You can also have groundwater inputs. In this situation where I'm talking today, those are going to be relatively minor, but in other places they might not be so minor. And then what goes on in the lake itself, there is a whole aquatic ecosystem. It includes not just diatoms, but bacteria, other types of algae, chrysophytes, some of the higher trophic levels of chironomids and cladocerans that eat the diatoms, rotifers, ostracods. All those processes go on in the lake. All those organisms are responding to these different processes in different ways, and the diatoms and other things that are preserved end up in the sediments. So the diatoms are taking in so if we've got wet and dry deposition of nitrogen coming in, and the diatoms respond to it because it's a nutrient, it makes them grow, we're going to see the result of atmospheric deposition from the atmosphere. So lake sediments are like tree rings. This is a section of a sediment core, and so it shows. This one actually shows annual layers from lake sediments of light and dark that have accumulated over time. And so we can come along and take samples of that material and process the diatoms within it and understand how have the species changed over time. And there's a lot of different ways that species can change over time, that ecosystems respond to different pressures. And if we look at ecosystem response, we can have different kinds of things that happen over time. You can have an individual response to some kind of pressure like nitrogen. And that can be in sort of changes in the amount of primary productivity, that there can be increases in growth. We can have changes at the species level in terms of ordering, which would be changes in abundance. And that's mostly what I'm going to talk about today. And we can also have changes in immigration, and that can be things like exotics, introduction of invasive species into various areas. So big deal. That stuff's going on, and but if we look at the bigger part of the western U.S., what we see is that the diatom species composition is changing at a certain threshold level. So we know that when there's this certain amount of nitrogen, it happens to be 1.5 kilograms per hectare per year, that triggers a change in species composition. I'm going to skip over this part and just go straight to some of the examples in the West before I talk about what we see in the Teton lakes. So here's a bunch of graphs. These are from two lakes. This is one lake in, well, they're both in Colorado, both from Rocky Mountain National Park. And just to orient you to these graphs, over on the left, we've got depth of the sediment core. So that's going from 0 to 20 centimeters. These are just little sediment cores. And then there's a bunch of different things plotted across the top. Let's see, the first thing I'll show you is over on the right is an age model. So we can use some different techniques to figure out how old are the sediments. And if we go over to this age model, we can see, it goes from, it's around, 1990. It goes back to the early 1700s in this sediment core. So we've got the history of the area. There's a line coming over at about 1950. And what we see is that there are these two species, Asterianella formosa and Fragillaria crotonensis, that here they are in this lake as well. And although they were present back hundreds of years ago, something happened that caused them to become much more abundant. And these might look like just little dark lines on a graph to you, but here there's a 60. So it went from being virtually nothing to composing 60% of the population was this diatom. So that's a very big change if you talk about something being a trace amount to really dominating the assemblage. And so this diatom this is what it is, Asterianella formosa. It's this one right here. Each one of these is a cell, and they're relatively large, and they form these beautiful colonies that the cells don't totally separate from each other when they divide. They form these spiraling colonies, and they actually are, this keeps them buoyant in the water column. It prevents them from sinking. So they're found in the plankton. They have a particular pH range that they like. But the big thing is they're characteristic of mesotrophic lakes. So lakes that have pretty good amount of nutrients present, pretty good amount of nitrogen present. And it is a marker of that. I'll go back here. The next thing I'm going to tell you about is the nitrogen and how we're tracking the nitrogen. We've got here something called delta 15N. And if I'm going to bump back over here and tell you about that. So that's, in order to look at the timing of these changes and the sources of nitrogen, we're looking at the nitrogen stable isotopes. So what that means is we're looking at, so nitrogen can come in different forms. It can come in a 14N, which is the normal form, or it can come in a heavier form, a 15N. And those ratios, those are naturally occurring. And they tell us something about the nitrogen biogeochemistry and the sources of nitrogen. So this notation simply tells us the ratio of N15 to N14 in any particular sample. And so we're looking at this atomic ratio of those two elements. So normally in air, the ratio is 0, but the activity of burning fossil fuels, agriculture, production of fertilizers is something that changes those ratios. And what we can see is in different processes make them go different ways. And it will be important to look at what are those and how do they change over time. So if we go back to that plot, we can see that this, the delta 15N started out somewhere around 4, and this is several hundred years ago, and then it was declining. So it's becoming depleted in N15. That also happened in the second lake. And I'll come back to the nitrogen too, but first I wanted to point out another thing that we see, another thing the lakes are recording. We've got lead. We can see that back in the early 1800s there were pretty significant concentrations in lead, and these are lakes in a national park. So really kind of far away from things we might think.
You said 1800s, you meant 1900s.
I said no, I mean 1800s, because is that what, am I not reading? Okay, 1900s, sorry, thank you. In the sort of really late 1800s, there was a lot of smelting that went on. Gold, silver, some of those operations for mining were pretty high up in the mountains, and they used lead to be able to obtain some of those minerals. And that lead, got up into the atmosphere, came back down in the lake, and we can see it there. And then it continued to increase up until about the 70s, and then it decreased. So what else carries lead? Gasoline, yeah, and it was, in the 70s, reduction to use unleaded gas. The lakes responded, it dropped again. So these high elevation lakes and other lakes too are seeing all that stuff happen and documenting it. So some of the other studies in the West said, well, maybe this change in nitrogen is only happening in the really high elevation rocky sites where there's not a lot of terrestrial vegetation. But here we see Asterianella formosa comes in here in 1980 or so, maybe even earlier. In other watersheds in Wyoming, here with a lot of terrestrial vegetation, Asterianella formosa still comes in recent sediments, and the nitrogen is also becoming more depleted at the same period of time of that change in Asterianella formosa. Same patterns in California, in the Sierra Nevada mountains. Sometimes the timing is a little bit different, but Asterianella formosa and this Fragillaria crotonensis come in around 1960, and this pattern is repeated. So we thought when we went to the Tetons that we would see really a very similar pattern, and we were looking for the appearance of Asterianella formosa to be able to mark some of these changes. This project in the Tetons was funded by the National Park Service. They wanted to know, are the lakes changing? Are the species changing because of atmospheric deposition of nitrogen? And so we screened a bunch of lakes, and we We selected them based on their concentration of nitrogen. Each one of these bars represents a different lake that we studied. The total nitrogen concentration in the lake is marked by this dark spot. So they range from a lot of nitrogen, 12 micromoles per liter, which is a lot in a lake, down to a relatively low amount. And the lakes also spanned the different speciation of nitrogen. So we had different amounts of total organic nitrogen, of particulate nitrogen, of nitrate, and ammonium. And it was really kind of nice that our lakes were all really quite different. And so we could be able to say of all these different type lakes, how do the diatoms respond to nitrogen? So I did this project with my colleagues, Alex Wolf from University of Alberta, who's a geochemist and also a diatomist. My postdoc, Megan Otu. Jill Barron, who's at USGS and Colorado State University. And she's really the person who has done the really important work on nitrogen throughout the West, and this is really her area of expertise. And then we had a lot of graduate student pack animals that came along to help us carry. Yes?
A couple slides back where you were showing the lakes, they didn't seem to be terribly far apart from each other in terms of miles, but you do have quite a bit of change in terms of the nitrogen between the lakes. Is that due to something like altitude or some being on the north side, say, of a range as opposed to the south side?
One of the things, that's a great question, and we wanted to have an elevational gradient. It was quite difficult to find an elevation gradient. They are quite close to each other. They differ in watershed size, but it doesn't seem that the change in nitrogen concentration is necessarily related to that, or their concentration differences between the lakes. But besides that, they're quite similar in terms of depth and exposure. That's a good question, though. We had Megan create an ingenious coring rig. We had lightweight rafts, inflatable rafts that we could carry up in packs, and a coring platform that we could put together in order to carry the equipment that we needed to drop into the lake to get cores of the sediments. This is what a gravity core looks like. It's a device that we simply lower down into the sediment. It has a bunch of weights tacked on, and so this goes in very gently, only as far as the weights will take it. And one of the nice things about this way of coring is that it is amazing at retaining the sediment-water interface. You can see the invertebrates that were sitting there on the surface. It's just not even disturbed. And that was very important to us because for us, the changes in the last decades are going to be occurring right here. You know, this is going to go back a few hundred years, but we really need to know with a very fine resolution what's happening at the surface sediments. So from the 7 lakes that we cored in the Tetons, in the first one, we thought, aha, Asterianella formosa, look, there's a certain time it used to, it was in the lake, but then it became more prevalent. But, it wasn't as much as the other sites. It was really only about a 20% of the population. But we do also see declining in delta 15N. And some changes in the other diatoms as well. There are other community level changes that were going on. In another lake, we did not see Asterianella formosa at all, but we saw changes in some of the other species. I haven't listed all the species. If I did that, it would be 150 going out that side of the graph. So I'm only showing you some of the more important ones here. So, we thought, well, something's happening with the diatoms, but we're not sure what. But the nitrogen is also changing, giving us the same indication that we have an anthropogenic signal. And the same with the other of the lakes. There's something changing, but it wasn't sort of jumping out at us. So we took the species composition, and probably some people will recognize this as being a ordination plot. So each one of these points represents the entire diatom assemblage scrunched down to 2 dimensions. So what this says, first of all, don't look at the size of the dots yet, just where they are, that the species were all kind of the same here, and then they were kind of different over there when you consider all of them. And so then the next thing we did was plot the nitrogen, the del 15N on top of that. And so the larger dots represent higher del 15N, and the smaller dots represent depleted form of N15. So we can see that, okay, in the older sediments where the diatom assemblages are the same, they're shifting in recent times. So that's one way of presenting that same plot. This is another way of looking at that same idea, which is here's the actual concentration or the actual N15, N14 ratio around 3, and then becoming very depleted somewhere near the 70s or 60s, and then this community response. So the diatoms, this is an ordination score. So it's simply a way of taking 150 species and saying how are they changing, and they are changing at that same time. Another way of looking at that is saying, okay, if we looked at that total variance in species, so those gray bars, what is it related to? Is it related to the nitrogen isotopic ratio, or is it related to the total nitrogen? So red is the del 15N, and black is total nitrogen. And we see that there are significant relationships between the diatom species, but we see that the diatom species are responding more to the del 15N than the total nitrogen. What is that about? Why would a species, so nitrogen is a nutrient, right? You need to take that in. So why would a diatom respond more to the human nitrogen than total bulk concentration? And I'll just leave that as a question right now and come back to that in a little bit. But we wanted to just confirm this a little bit more. I did another analysis of testing the difference between historical values and modern values. So took 1960 as an arbitrary break. I wanted to test the significance of this change. Prior to 1960, there was one group of the way that a diatom assemblage responded. After 1960, it goes off on a different trajectory. And this trajectory is using something called a relate test. Before 1960, it's a random pattern, and that's highly significant. After 1960, it's not random, and in fact, it's directional. And what I'm looking at is this bar is a test of the significance in the relate test. So all the lakes do that. So before 1960, they're random. After 1960, they're directional. And we saw that in all the lakes. So the story is all the lakes are showing this directional change in relation to N15. And that change may not be so much how am I going to say this, not garbling it. We think that what's coming in the human anthropogenic form of nitrogen is more enriched in ammonium, so that the diatoms can take it up more readily and respond, and that's part of what we're seeing in these lakes. And so this response is happening here over time. We're seeing a species reordering. We did not see changes in productivity, and we're not seeing right now introductions of species. So this anthropogenic signal is of decreasing delta 15N is happening in all the lakes. There's biotic response that is somewhat different than other studies. And in that the entire community is changing rather than just one or two. So I'll just leave it at that. Give thanks to my many collaborators and students and funding agencies that gave support to this project. And I would be glad to answer any questions.
Got that. Bye.